Exploring Life. Chapter 16, Exploring Evolution. Part 1. Let's start today's journey with a simple concept. What is a population? In biology, a population is a group of individuals from the same species, living in the same area at the same time. Think of the town or neighborhood you live in. That's an example of a local human population. Now, let's introduce the idea of the gene pool. The gene pool includes all the alleles present in that population, whether or not they're currently expressed. Even if an allele isn't currently being expressed in anyone, it's still part of the pool. Where do new alleles come from? As we previously discussed, they arise through small-scale mutations, small changes in DNA, and they're constantly being reshuffled through the process of sexual reproduction. This includes mechanisms like crossing over, independent assortment, and random fertilization. Population genetics is the branch of biology that studies genetic variation within populations. We're evaluating two alleles in a population, the dominant allele, P, and the recessive allele, Q. We can still use a Punnett square to visualize each genetic cross, just like before. We place the potential alleles, P and Q, on the sides and determine the outcomes in the intersecting boxes. What you're seeing here is just one of 9 possible parental combinations, accounting separately for male and female contributions, shown simply to illustrate the idea. Now, imagine combining all the individual Punnett squares possible across the entire population into one larger, unified square. This combined Punnett square now represents the overall genetic structure of the population. The top left cell, labeled P times P, represents the frequency of homozygous dominant individuals, P squared. The bottom right cell, Q times Q, gives us the frequency of homozygous recessive individuals, Q squared. The remaining 2 cells reflect heterozygous individuals, represented by 2pq. So it's the exact same Punnett square logic, just scaled up to the level of an entire population. Homozygous recessive individuals often have a distinct, visible phenotype, something we can directly observe in a population. And from that single known value, we can calculate the frequencies of alleles and genotypes. In other words, just by observing one trait, we can estimate the entire genetic structure of a population. Did that sink in? Great. Let's walk through it step by step. Let's imagine we have a population of 200 rabbits. Out of those, 72 have white fur and 128 have brown fur. We know that white fur is a recessive trait, which means the genotype of the white rabbits must be B lowercase. So, 72 out of 200 rabbits, or 36% of the population, have the B lowercase genotype. Now we determine the allele frequencies for this population. Since 36% of the individuals are B lowercase, we know the frequency of the B lowercase genotype is 0.36. Taking the square root of 0.36 gives us 0.6, or 60%. This is the frequency of the recessive allele B. To find the frequency of the dominant allele B capital, we subtract 0.6 from 1, giving us 0.4, or 40%. Together, these values give us a clear snapshot of the gene pool in this rabbit population. Now let's calculate genotype frequencies. Starting with B lowercase squared, it's already given as 0.36, or 36% of the population. For B capital, that's B capital squared, 0.4 squared, which equals 0.16. That means 16% of the population is homozygous dominant. And for B capital B lowercase, we have a couple of ways to calculate it. The simplest is to subtract the frequencies of B lowercase and B capital from 1. That gives us 0.48, or 48% of the population, the heterozygous individuals. Let's say that, after a while, the frequency of fur colors changes in the rabbit population, and white rabbits become more common. You can repeat the same simple steps we just used to calculate the new allele frequencies. If you're dealing with a wild population, the change you observe reflects evolutionary forces at work, a process known as microevolution. Microevolution refers to small-scale changes in allele frequencies within a population over time, changes that we can observe across generations. The strict conditions under which a population would remain unchanged are described by the Hardy-Weinberg principle. These conditions include no natural selection, meaning all traits contribute equally to survival and reproduction. A very large population, so chance events don't have a major impact. No migration, meaning no new alleles enter or leave the population. No mutations, so the gene pool remains stable. And random mating, with no preferences for particular traits. As you can probably tell, it's nearly impossible for a population not to evolve. One of the conditions is no mutations, and as you already know, mutations happen all the time. So instead of expecting Hardy-Weinberg equilibrium in the real world, we use this principle as a baseline. The greater the deviation from these conditions, the faster the allele frequencies change, and the more pronounced the evolution becomes. Microevolutionary forces shape the genetic makeup of populations from one generation to the next, and each is aligned with the Hardy-Weinberg principle it violates. So, what are these forces? Natural selection, where advantageous traits become more common over time. Genetic drift, random changes in allele frequencies, especially in small populations. Gene flow, when individuals migrate between populations, introducing new alleles. Mutations, spontaneous changes in DNA that create genetic variation. And non-random mating, which can increase the frequency of certain traits. We'll explore each of these in more detail, leaving natural selection for last. Genetic drift refers to random changes in allele frequencies from one generation to the next. These shifts aren't driven by natural selection, they happen purely by chance. Genetic drift has the strongest impact in small populations, where random events can have outsized effects. Over time, drift can lead to dramatic outcomes. Some alleles may disappear entirely, while others may become fixed, meaning they reach 100% frequency in the population. Two other important forces that drive microevolution are gene flow and mutation. Gene flow, also called migration, occurs when individuals move between populations. As they arrive, they bring new alleles, or remove existing ones, changing the genetic makeup of each group. Mutations, on the other hand, are spontaneous changes in DNA. Some of these changes create entirely new alleles. Over time, these rare but crucial mutations fuel genetic variation, the raw material that evolution works with. In real-world populations, including humans, mating is rarely random. In many populations, individuals choose mates based on specific traits, a process known as non-random mating. For example, in assortative mating, individuals may prefer partners with similar characteristics, like size, color, or behavior. Geographical proximity also plays a major role. When populations are isolated, mating tends to happen within a limited group. And as we've already seen, inbreeding within such groups increases the likelihood of homozygous traits appearing, sometimes leading to the expression of harmful recessive alleles, like Tay-Sachs disease, which we discussed in lecture on applied genetics. And only now, we arrive at natural selection. Unlike the other forces we've discussed, natural selection directly drives changes in allele frequencies. Traits that improve survival or reproduction become more common over time, while less favorable traits are gradually weeded out. This is how populations adapt to their environment, not by chance, but by differential success. Natural selection can act on traits in different ways, depending on environmental pressures. There are three main modes of selection: directional, stabilizing, and diversifying. On this image, the original population's trait distribution is shown in the top. Right below it is directional selection. This mode favors one extreme of a trait. For example, if larger body size offers a survival advantage, individuals with that trait are more likely to survive and reproduce, shifting the average size upward over time. Stabilizing selection is shown on the top. Here, the average trait is favored, while extremes are selected against. A classic example is human birth weight. Both very low and very high birth weights can pose risks, so intermediate weights are more likely to survive. Finally, diversifying or disruptive selection appears in the bottom. It favors both extremes over the average. For example, in some bird populations, individuals with either very small or very large beaks may have better access to food, while those with medium-sized beaks are at a disadvantage. Natural selection does not act alone. Variation and sexual reproduction are essential. Mutations produce the genetic variation, and selection shapes it. Without mutations introducing new alleles, selection would have nothing to act on. As you can see in the image, with each round of selection, certain colors disappear while others are added. The population shifts, and the base color gradually changes. This is evolution in action. Small genetic changes, shaped by selection over generations, gradually shift which traits are common in the population. Evolution never acts on a single species alone, it's almost always coevolution. Coevolution occurs when species evolve in response to one another, shaping each other over time. Pollinators and flowers, or predators and prey, are classic examples. When one changes, the other adapts. But even these are oversimplifications. In reality, coevolution is not just a two-way street. It's a dynamic, multi-species network, an entire web of organisms living together, depending on one another, and constantly interacting in complex ways. Before we dive into macroevolution, which includes the origin of species and other large-scale patterns, we need to review what a species actually is. At first glance, defining a species might seem simple. But in biology, it's surprisingly complex. There's no single, universal definition of species. In fact, biologists have proposed over 20 different species concepts, each useful in different situations. In this course, we'll focus on four major ones: the biological, morphological, ecological, and genetic species concepts. The biological species concept is the dominant species concept in biology because it's clear and unambiguous. If individuals can interbreed and produce fertile offspring, they're considered the same species. But it doesn't always work. It breaks down for asexual organisms, which don't reproduce through mating. And even for sexually reproducing organisms, reproduction is often difficult to trace. It's nearly impossible to observe mating patterns in the wild, especially across multiple generations, and many organisms won't breed in captivity. It also doesn't apply to fossils, because, plainly put, they're dead. So unless you're studying something common and easily observed, you'll need to rely on other species concepts to make sense of what you're seeing. The morphological species concept defines species based on physical characteristics, essentially, what organisms look like. This includes features like body shape, size, coloration, and other anatomical details. The underlying assumption is that if two organisms look different enough, they probably aren't mating. This concept is especially useful when reproductive data isn't available, such as with fossils or preserved specimens. In paleontology, it gives rise to the idea of a chronospecies, a species that changes gradually over time. Each generation may differ only slightly from the one before it, but over long time spans, these small changes can accumulate into a clearly distinct form. Because the transition is continuous, all intermediate forms are usually grouped into a single species. However, this concept also creates major hiccups. Juvenile animals often look very different from adults. For example, a juvenile T-rex was once mistaken for a separate species due to its small size and odd proportions. It also overlooks natural variation within a species. Strictly applying this concept today might give us dozens of species of dogs, cabbages, or pigeons just based on shape, size, or color, despite them being genetically the same. And then there's sexual dimorphism, when males and females look dramatically different. An extreme case? The deep-sea anglerfish. The female is large and lures prey with a glowing light. The male is tiny and fuses permanently to her body, living off her nutrients while supplying sperm. For a long time, he was classified as a parasite, an easy mistake. He kind of is, but still, they're the same species. In ecological research, it's often impossible to confirm whether groups are actually interbreeding, especially across generations. But here's a key insight. If two groups never meet in nature, they don't breed. That's why the ecological species concept defines species based on their ecological niche, the unique role each group plays in its environment. This includes where they live, what they eat, how they behave, and how they interact with others. A real-world example comes from African cichlids. In the same lake, look-alike cichlid species live at different depths and occupy distinct feeding zones. Even though they appear similar, their niche separation prevents interaction and can lead to ecological speciation. Of course, the ecological species concept has limitations. It can oversplit species based on lifestyle differences. Take humans, we live in vastly different environments, eat different foods, and follow diverse cultural behaviors, yet we're all one species. Same with orcas, some groups hunt fish, others hunt mammals. They use different sounds and live in different regions, yet they're all Ursinus orca. So while this concept is useful, it's definitely not universal. At first glance, the genetic species concept seems like the answer to everything. We've finally moved beyond breeding, we're focusing entirely on DNA. This concept defines species based on genetic differences. If two populations have distinct enough DNA sequences and show limited gene flow, they're considered separate species. It's especially powerful for identifying cryptic species, organisms that look nearly identical, but are genetically distinct. But here's the major challenge. Where do we draw the line? There's no universal threshold for how much genetic difference, or in which part of the genome, is enough to define a species. There's no universal threshold for how much genetic difference exists between recognized species. The answer depends on the organism, the genes being analyzed, and the methods used. DNA comparisons show just how tricky this can be. Humans and chimpanzees, two clearly separate species, share over 98% of their DNA. Yet some prokaryotes can differ by 15% or more genetically and still be classified as the same species. So while the genetic species concept is a powerful and modern tool, it's definitely not a one-size-fits-all solution, at least not yet. In the second part of the lecture, we'll examine how new species arise, how major biological innovations emerge, and how evolutionary relationships are traced through time. Now it's time to pause and reflect. How well do you understand the key concepts we've covered? Take a moment to review your notes, quiz yourself, or revisit any sections that were unclear. Self-evaluation is a powerful tool to strengthen your understanding and retention.